Okay, we're going to go through what y'all need to study for your state test, which is Tuesday, May the 15th. We're going to go through competency by competency, just hit the high points of things that we need to go over and review and make sure that we know. Okay, so competency number one is inquiry, and that means we're looking at scientific method. And so when we look at scientific method as a whole, we're just looking at the way that we conduct a good experiment. Our experiments always begin with a hypothesis. This is just a prediction. It's in an if-then format. So, if it's dark and cloudy outside, then it's going to rain. This hypothesis contains our two variables. So the independent variable, which is the variable that I can change, is going to follow the word if, and then the dependent variable is going to follow the word then. And so when we look at these experiments, it's not just about setting them up. We need to make sure that the experiments are valid and that the results are results that we can trust. And we also need to make sure that we can obtain good results and really read and understand those results. So good experiments, remember, have an experimental group and they have a control group, and they only have one independent variable. In that control group, you do nothing to it, okay? So at the beginning of the year, we did a fertilizer experiment on our plants, and your group planted either bean or corn seeds. So, so you planted a bean in a cup that had soil that you did not include fertilizer in, and then you had a bean that you planted in a cup with soil that did contain fertilizer. And so remember, the fertilizer was the only thing that was different in those groups, and so that was our independent variable. The outcome was the dependent variable. So in that case, we were measuring growth. And so the growth changed based on whether or not we had fertilizer in that group, okay? When we chart our results, remember, we're going to use graphs. And the two types of graphs that we use most often are bar graphs and line graphs. Bar graphs are just comparing groups of data, and the line graph is showing how one thing is changing over time. And so bar graphs, you may have more than one thing included in it. When we look at graphs, we're always going to have our dependent variable on the x-axis and the independent variable on the y-axis. Down here we have a graph, and this is just a line graph, and we have our independent variable down here on the x-axis and our dependent variable on the y-axis, okay? And so if we look at this graph, it's showing us several things. We have a title up here. It tells us the number of people in a store. And so over here we have the number of people, and here we have the time, okay? And so when we look at our particular graph, we can see that if we're trying to see when we have the most people in the store, we're just going to look for the highest point on the graph and make sure that you're reading the scale correctly. So this is, of course, 20 here, and this is 25, so it'd be 21, 22, 23, 24, 25. So we have 22 people here, so that would be, the peak point or time in that store. And also just make sure that you notice that you have a good scale down here and that you're reading your graph carefully. When you have questions with graphs, look at them first because they're a picture and they can oftentimes tell you what the question is going to be about. Many of our questions also have charts, and so like this, or a data table if you want to call it. And just be careful when you're reading the chart. Look at it carefully. Again, it's going to tell you what's in it. So we can see just from this, we have 7 different plant groups. We have soil pHs recorded for each of those groups, and then we have plant growth, okay? So this is obviously an experiment where they tested how the pH of the soil would affect the growth of the plant. So if we look at what pH would be best, obviously we're wanting to see the most growth. So we'll just scroll down through this table, and we see that for 6.6 and 6.8, we have 53.3 centimeters. So if we were asked what would the optimum pH be for this, we would say either 6.6 or 6.8, and that would be somewhere in our answer choices. If they ask us what would be the worst pH for the soil, we would just look here and find our lowest number, which you see is 22.9, and we would see that that would be a pH of 7.2, okay? Now remember, 7 is a neutral pH, and everything that's below 7 is going to be an acid, and everything that's above 7 is going to be a base. And of course, the further away you get from 7, that would be a stronger acid or stronger base. And we'll talk about that more in just a minute. If we look, at other pictures and we look at how these experiments are set up, it is possible to have more than one experimental group as long as you're testing the same variable, okay? So in this, we're testing the effect of water acidity on coral bleaching. And so, you know, what coral is, you know, coral reefs and things like that. It's a type of cnidarian, which lives out in the ocean. It's an animal. So we've got normal seawater here at 8.2, which is a mild base. We're seeing what happens after 8 weeks, and we're seeing that 10% of our coral was bleached after that time, okay? Now over here, we have 2 experimental groups. And again, since we're still testing pH, it's okay that we have 2 groups. This one is at pH 7.9, and this is at pH 7.65, okay? We're still keeping everything the same. We have 1, 2, 3, 4, 5 particles here, 5 here, 5 here. We have same apparatus, same amount of water. The only thing that's different here is the pH. And so once we obtain our results, we can compare these to each other. Now, if we look at validity, which means how well can we trust our results, there's several factors that we must have. So we do have to have control and experimental groups. And up here we do. We have normal seawater, which is our control group. We also should only have one variable, which in that previous slide was pH. We need to have everything listed out very clearly and make sure that we have the same everything in all groups and in same amounts of everything. So in this experiment we had same amount of water, same apparatus. We keep them in the same environment, the same number of coral pieces and whatnot. To increase your validity, you want to repeat your experiment. The more you do something and the more you get the same outcome, the more you can trust the results. And also you'll know if something went wrong if you have that one, set of results that comes out different than all of the others. And you also want to test a lot of individuals. You want to have what's called a large sample size. So up here, we only tested 5 corals. Well, it would have been even better if we had tested 10 or if we had tested 15 or 25 even, okay? Down in this experiment, we just have a little question, and it's asking us to tell them what we're trying to answer. What are we testing? And so it tells us in the question, a student will measure and record the growth of 2 flowering plants every other day for 10 days. And so you can see we have identical plants. It tells us we're adding 200 mils of water to each pot. But in this pot, we seem to put the fertilizer. So just think for a minute what our independent variable would be and how we would construct a hypothesis. So if I was conducting this experiment, I would say if 10 milliliters of fertilizer is added to pot 1, then pot 1 will show more growth. And so our independent variable would be the fertilizer, and the dependent variable would be the growth, okay? So our question says what is being tested, and so our answer here would be C. Does fertilizer added to the soil lead to taller flowering plants? And the next question would be, what could we do to increase the validity of this experiment? And so earlier we talked about we needed a number of subjects. And so I wouldn't just test one plant with fertilizer and one without fertilizer. I would test 10 plants with fertilizer and 10 plants without fertilizer, okay? And then maybe I would repeat this experiment, after I went through this, I would do it over again, and that would increase my validity. So the more flowers that I had in each group, the better my results would be. So just keep those things in mind when you're looking at your experiments. Don't forget about microscopes. And the part of the microscope that everyone seems to not understand is what the diaphragm is. The diaphragm is located right under here, kind of where my pointer is. It's the part that adjusts the amount of light that's coming into the specimen. And then sometimes we also get asked how to calculate total magnification, which is when you look through the microscope up here, how much are you really magnifying what's on your slide down here? And don't forget that there's a lens up here in the eyepiece, and then there are these lenses down here. So the eyepiece has a magnification power of 10, and if you're using the 45 objective down here on the bottom, you multiply those two numbers together, and you would say that you're magnifying 450 times, okay? Last thing are your safety symbols. Just remember the difference really in these two are the ones we tend to get mixed up, radioactive and biohazard. We see these biohazard symbols on the containers, the red containers in our doctor's offices where they put the shot needles after they give us an injection. And I think about shot needles being really sharp and pointy. And so that helps me to remember that this is the biohazard safety symbol. I think all the other ones you could pretty much pick out. And then for lab safety rules, remember the number one thing you can do if something happens is to tell the teacher. And also, if you're looking at protective gear, the most important thing that you can protect on your body is your eyes. So if they ask you about aprons versus goggles, always say that you need your goggles first because that's going to protect your eyes, okay? All right, we're going to move on to competency 2, which is chemistry. And for this one, we're going to look at bonds, macromolecules, enzymes, pH, and then our 2 life reactions, photosynthesis and respiration. There are 3 big types of bonds that we've learned about this year. The first one is the hydrogen bond, and that's going to be found right here between different molecules of water. It is also found in DNA when we look at how adenine bonds to thymine. So remember our A and T go together and our C and G go together. Well, the bond that holds A to T together, it's a hydrogen bond as well. But most often we get asked about the hydrogen bond here. In covalent bonds, you share, and ionic bonds, you transfer, okay? And so it's just like a gift-giving relationship. So, if mom and dad, buy a car for you and a sibling to share, then you're going to have the car one day, and then maybe your sibling would have it the other day, okay? And that's how these covalent bonds work. They're going to share the electron. So one gets it sometimes, and then the other gets it the other time. Ionic bonds is a total transfer. It's all yours. So, if I give you a pencil, I'm giving it to you, and I don't expect for you to give it back to me, okay? And so that's a little bit different. And the most common example is sodium chloride, which is table salt. So down here we have some pictures of ionic bonds. When you see ionic bonds drawn out, typically the circles don't touch, like they don't touch here. And you'll also a lot of times see an arrow, and you can count up the number of electrons. So, the outer shell fills up with 8, right? So fluorine over here has 2, 4, 6, 7 valence electrons, so it really wants one extra one. And since lithium only has 1 in the outer shell, it wants to give it to it. So this is an ionic bond. Then, of course, this is an ionic bond as well. It's the same situation. Chlorine has 7 valence electrons. It wants 1 more to fill up. And remember, it wants it really bad. Sodium has it, so it gives it to chlorine. And that, again, is the ionic bond. Back up here with the covalent bond, you saw the circles touch. So remember, if you share something with somebody, you have to be close to them. So covalent, your circles are probably going to touch. Ionic, they probably are not, and you'll also see arrows. The next thing that we need to look at is water. And water, it truly is the elixir of life. All life has to occur in water, so fertilization occurs in water. Water transports things around a cell because the cytoplasm is made of water. So water's got some really unique properties that allow things to be alive, really. So when we look at those properties, the number one property is polarity, and that means that water has a positive end and a negative end. So the end where the hydrogens are positive, and the end where the oxygen is negative. Cohesion means that water sticks to itself. Okay, so co is together. Adhesion means that water sticks to other stuff. Okay, so glue, for example, is an adhesive. So if you were going to buy glue, at Home Depot or Lowe's, you would go to the adhesive aisle, because glue is going to allow you to stick two different things together. And so when you think about adhesion, think about water sticking to other things. Water is a good solvent because it's positive on one end and negative on the other. We know that when water freezes, it gets less dense. Okay, so that means it's going to float. Dense things always sink to the bottom. Less dense things don't. So water has a low density and it floats. And that's good for areas where it's really cold, so it can preserve aquatic wildlife that lives under the ice and keeps them from dying. People do go ice fishing where they drill holes in the ice and they stick their lines in, and they actually are able to catch something. Water also has a high specific heat, which means it does not want to change temperature. And so that is why the temperatures on Earth remain relatively constant all the time. And it's also why our bodies can hold a constant temperature even if we go outside and it's really hot, or if we go outside and it's really cold. So our body temperature is going to stay about 98.6, for the most part, unless we're objected to something really extreme. And then the high heat of vaporization means the same thing. So we all know that steam is hotter than water because the molecules, of course, are moving a lot faster when it's a gas. And this is also goes right in hand with this. Water does not like to change temperature at all. Okay. When we look at pH, water is a neutral substance, and that means that water has exactly the same amount of hydrogen ions and hydroxide ions. Okay, so if you have the same hydrogens and hydroxides, your solution is neutral. But when we add other things into water, we can change the pH. So if we add something into water and extra hydrogens are released and we have more hydrogens, then we have an acid. If we put something in water and we end up with more hydroxides, then we have a base. And so the pH scale tells us what we have. So 7 is neutral. Anything above 7 is a base. Below 7 is an acid. Okay, so here's our pH scale. And remember, every time we move on this scale, it's a times 10. So if I'm going to compare water to baking soda, okay, so water 7, soda is 9. Baking soda is going to be 10, 100 times more basic than water. And so what that means also is that baking soda is going to contain 100 times more hydroxides than water. Okay? So if I compare water to vinegar, we know vinegar is going to have more hydrogen, right? So we go 10, 100, 1,000, then it's 10,000. So vinegar has 10,000 more hydrogens than water. Okay? So remember, every time you move, it's times 10. If we have a solution and we want to neutralize it, we can use a buffer. So if we have an acid and we want to neutralize it, we can add a base to it. Okay? And so what that buffer is going to do, so if we have an acid, which has a lot of hydrogen, when you add the base, which is the buffer, it's going to absorb some of the hydrogen. Okay? So since acids have too much hydrogen, the buffer is going to absorb some hydrogen. Okay? And if you have a base and you don't have enough hydrogens and you have too much hydroxides, then your buffer is going to put hydrogen out. So you just kind of think of where you are. You just got to remember that acids have more hydrogens and bases have more hydroxides. Okay? The next big thing that we need to talk about are enzymes, which you know are absolutely the most important type of protein that we have. Enzymes control our chemical reactions. And enzymes, remember, they're not changed by our reactions. So down here, the enzyme is purple and the substrate is this yellow thing. And remember, the substrate fits into the enzyme just like a key fits into a lock. And we only have so many enzymes, so that means that we can use them over and over again. It doesn't take much enzyme to help out the many reactions that we have in our bodies. And remember, our enzyme graphs, sometimes they'll go up and then they'll level off and they'll kind of get straight. And that means that our enzymes are all being used at the same time. Again, it's the shape of the enzyme that matters. This little place, this little notch on the enzyme is called the active site. That's where the substrate fits in. Enzymes, remember, are affected by pH and temperature and salt. So if the enzyme gets too hot or if it gets put in acid, then this active site will get messed up and the substrate won't fit into it anymore. And so that's what's going to mess up the reaction. Okay. Next thing, we need to know our macromolecules, and there are 4 categories of those: carbs, lipids, proteins, and nucleic acids. And you need to know an example and what the monomers are, okay, in addition to the function. So carbs and lipids both give you energy. Both of these are made of carbon, hydrogen, and oxygen. But in the carbohydrate, if you count up here, you're going to see that there's a 1 to 2 to 1 ratio of the carbon to hydrogen to oxygen in this in carbohydrates. Carbs, of course, are used for energy. There are many different types of them. The cell wall of plants, that's even a carbohydrate. Okay, so carbohydrates are carbon, hydrogen, and oxygen, 1 to 2 to 1 ratio. Their function is to store energy, short-term energy, and their monomer is the monosaccharide, which would be glucose, fructose, and galactose. Lipids, on the other hand, they're long-term energy storage molecules. Hormones and steroids also fall under the lipid category. When you see a picture of lipids, you're going to see lots of hydrogen, lots of carbon, but not a lot of oxygen. The monomers are glycerol and fatty acids. Okay. Proteins are made of carbon, hydrogen, oxygen, and this time we also add nitrogen. They are used for, many of them, for structure. We just talked about enzymes. The monomer of proteins are amino acids. So this is what an amino acid looks like. So remember, if you're looking at a picture, you're looking for the C, H, O, and N only. All of our enzymes, remember, are going to end in ASE. And then finally, your nucleic acids, they store information. So examples are DNA and RNA and even ATP. Nucleic acids are made of C, H, O, N, and P. So you've got carbon, hydrogen, oxygen, nitrogen, and phosphorus. And again, they store information. We also need to know what DNA looks like. So you need to make sure that you can recognize a picture of DNA. You need to know that A and T go together, that C and G go together, all those good things. And you need to know that the monomers of that is the nucleotide. Okay? For photosynthesis, we need to know what goes in and what comes out. And so sometimes y'all start answering questions really fast and you get these mixed up. So just think carefully when you see a question about photosynthesis. Remember, what's going in is carbon dioxide and water and sun. And what's coming out is glucose and oxygen. Okay? Photosynthesis happens in the chloroplast. Plants do it, but there are also a whole bunch of protists that can do it too. The majority of the chloroplasts are in the leaves of the plant. And so when plants put on a lot of leaves like they're doing now, photosynthesis is, really going crazy. It's kicked into high gear, okay? And of course, photosynthesis makes our oxygen. When we think about how the carbon dioxide comes in to the plant, it comes in through these holes called the stomata, which is down here at the bottom. The stomata also allow the oxygen to escape. Now, water can also leave through the leaves of the plant. Remember, that's called transpiration, and we'll talk about that in just a second. When you think about where the water comes in for photosynthesis, it comes in through the roots, okay? And so since this is a cross section of a leaf, we also have our vascular tissues here, and this right here, this blue, is representing your xylem, which is the tissue that transports the water for us. Okay. Here's a picture of transpiration. You know, water for photosynthesis comes in through the roots. It's going to be pulled up through the xylem by a process called capillary action. And that means that water is sticking to the edges of the tubes of xylem. All of this is speeded up when water evaporates from the leaves through transpiration. And so remember, temperature and wind can speed up transpiration, and then of course humidity slows it down. Respiration is the opposite of photosynthesis, and it happens in the mitochondria. So for respiration, we need glucose and oxygen, and we're going to get out, I'm supposed to say O2, we're going to get out water and carbon dioxide. So what comes out of your lungs is carbon dioxide, okay? The main thing that we're going to get is, you know, this range of 36, it's also 36 to 38 ATPs from the reaction. That's what we're going to use for our energy, okay? This is just a generalized picture of respiration. Remember, it starts with glycolysis. Then if you have oxygen, you can go and do Krebs and electron transport chain, okay? When you look at respiration, it can be done in two ways. It can be done aerobically, which means you have plenty of oxygen, and you're doing that in the mitochondria, or it can occur anaerobically, which means you don't have any oxygen, and that's going to occur in the cytoplasm. Okay, so what happens is cells that need a lot of ATP consequently also need a lot of oxygen. So muscle cells are a good example of that. If our muscle cells temporarily run out of oxygen, they'll switch over and do fermentation. Okay, and in us, that's going to be lactic acid fermentation. Lactic acid is what makes your muscles sore after you've done a lot of exercising. Okay, plants and yeast can do the same thing, but they're going to do alcoholic fermentation. So they're going to produce alcohol and carbon dioxide as their waste product here of this process. So that's how beer and wine, everything over at the liquor store is made is through alcoholic fermentation, where we're doing lactic acid fermentation. Once we get the ATP, we have got to extract the energy from it. So ATP stands for adenosine triphosphate. So you have 3 phosphate groups here. This right here, this little burst represents the energy, and what we've got to do is take off this 3rd phosphate group to get the burst of energy, okay? And the way that happens is by adding water. So in order for us to get energy out of ATP, we've got to have a lot of water. We've got to be well hydrated. And so water helps us out, and that's called a hydrolysis reaction. Moving on to Competency 3. Competency 3 is basically ecology. You've got to know characteristics of your biomes. You need to pick those out. You need to know what kind of stuff lives in the biomes. And then you've got to be able to identify biotic and abiotic factors. Okay? Ecology means the relationship between organisms and their environment. So an organism is simply one thing. Okay? If you have a group of the same species, the same organisms, that's what we call a population. When we mix different populations together, we get a community. So here you see you have more different types of organisms here in our community. Then you put different communities together, including the biotic and abiotic factors, and you get your ecosystem. Then you put all the ecosystems together and you get the biosphere, which is just basically the whole wide world. The ecosystem includes the biotic and abiotic stuff. Biotic means it's living, abiotic means it's not, and it never was, okay? So if you look even at things like feathers or bones, that's biotic because they came from something living. Abiotic things include factors like the amount of water, the amount of rainfall, snowfall, whatever, the soil and its content, the temperature, things like that are abiotic. So, rocks and things like that are abiotic, things that are non-living. A biome is just a huge, huge area, and it's determined and defined by what lives there. And the things that live in a biome have to be able to tolerate the conditions of the biome, which the two most important ones are temperature and rainfall. Okay. Here's a pyramid of the 6 major biomes that we have. So down here we have moving from wet to dry. So we have decreasing moisture content, and up here we have decreasing temperature. So the wettest and the hottest is the rainforest, and it's the most diverse. Remember, the soil is terrible. The organisms that live here have shallow roots. We have lots of things that like to climb that live in the rainforest because there's so many trees. There's the huge canopy. It's a great way for them to escape predators and things like that. Flowers that are found in the rainforest are really bright, colorful, and pretty to attract pollinators because there's so many bright and pretty things. Okay. The next biome under that with regard to diversity, so the second most diverse one is the temperate deciduous forest, which is our biome. So we are not quite as hot, not quite as wet as the rainforest. We do have 4 seasons, which is something that we need to know about our biome. So that means we have lots of diversity. Deciduous trees lose their leaves, and so when all that organic matter, falls off of the trees and it starts to decompose, it replenishes our soil. So we have super nice, nutrient-rich soil. We have lots of woods. We have lots of open land. So we have many, many different organisms living in our biome. Okay. If we look at grasslands, remember grasslands are going to be a lot drier than our climate. So you don't see trees here, and so that's why we're going to have big herbivores that live on the grasslands. You're not going to have as many climbers and things like that, so you won't find as many squirrels and things like that per se in the grasslands. You'll find more prairie dogs and organisms like that can burrow and live underground just because you don't have the trees. It's also going to be really windy, which is going to make our grassland drier than our temperate forest because remember, trees block a lot of wind. So whatever lives here has to be able to tolerate that wind. The difference in the grasslands in the U.S. and the savannas is, of course, that the savannas, they're going to be a little bit hotter than the grasslands that we have here. They do have a few more trees that live there, and so consequently you've got some larger herbivores that can survive there, or more variety in those. Then, of course, the deserts, no trees, sand is the soil. Plants that live there are going to have super deep roots to get to the groundwater. You're not going to have big organisms that live there just because there's not enough water to sustain them. So animals there, think about all the rattlesnakes you've seen in the desert, little desert rats. You're going to have, some, big birds and things like that, vultures and whatnot that can live there. But it's just going to be really hot, really dry. But remember, your temperature lines may look kind of weird for the desert because it's super hot during the day, but it's also really cold at night because they don't have any humidity. Okay. Taiga is your coniferous forest, so you're going to have evergreen trees that live there. It's not going to be that wet, but it is wet enough where you can grow the trees. It's going to be cold. Okay, so you're not going to have oak trees and maple trees and things that live there in the taiga. And then the tundra is kind of the desert of the north. So in the true tundra, you're not going to see trees. You're going to have permafrost as your soil. So remember, permafrost is frozen ground. The ground doesn't thaw out very often, so you'll have, maybe some grass, maybe only moss and things like that live, way up in the tundra. So stuff that lives in the tundra has got to be able to deal with the cold temperatures. So they're going to be fat and they're going to be furry, or they're going to migrate, or they're going to hibernate out of there because of the extreme conditions. Okay. Competency 4 is cells, so you've got to look at organization. You've got to know the difference in prokaryotes and eukaryotes. You've got to know the functions of your cell organelles, cell division, and how stuff moves across a membrane. Okay? So the 2 types of cells that we have are prokaryotes and eukaryotes. Prokaryotes are bacteria. They don't have a nucleus, they don't have organelles, but they do have ribosomes. Okay? They also do have a cell wall. They do have cell membrane. Their DNA is circular, but it's not in a nucleus. Okay? Eukaryotic cells are larger. They're going to have a nucleus. It's plant cells, animal cells. Don't forget protists. And fungi cells are also in this particular category. I'm going to pause so I can charge my